Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers back home, welcome back again, and here we are with the other part as a continuation of protein synthesis, which actually has two major components as the major steps. Remember, we talked about the genetic code, the coded information, our dictionary and stuff like that, the ATM and all those things we've been talking about, referring as to how actually what we look as being invisible can finally turn up into a structure. But the question is, how does it all work out? When we were talking about vision impairment, and we were looking at some of our members in the learning community unable to do the proper writing as we do, but how come that they are also able to sit exams and they still do the same exams as you do? How do they write their exams? How do the teachers get a chance to be in position to have access to their scripts and still mark them and get scores? And they do so well. So, this teacher comes and picks what this other student has done by poking through the paper. I remember that maybe one small dot on top of the small square and another small dot at the bottom of the square and another one on the right was represented as a certain letter of the alphabet. I don't know if it was A or B or C, something like that. So those three dots in a small square, the person, the teacher responsible for transcribing this information into letter A would have those skills. So I can't read the three dots, but I'm able to read the letter A. So what does this teacher do? He has to transcribe the script for this vision impaired student into a form that I'm able to read. So we call that transcribing. And then finally, when I have the letter, that's a translation. So in today's lesson, we are going into this genetic code of ours and pick out the information that is hidden here, the invisible, the codes, extract them, pull them out, take them somewhere they should be in position to be read. So the supervisor will collect these papers from the learner and then put them in an envelope and then pass them to REB. Then REB officers will call this other teacher who is in position to be able to read those dots and lines and da-da-da-da-da for that vision impaired student and then start doing the transcribing. Because those are codes a student has done in the exam, the vision impaired students. So this teacher will come and read and look at those codes and decode them into a format. So, one, we only need to make a simple outline of how we have our hair, how we have our muscle, how we have our nails, that we don't need to start adding others in the salons. No, you naturally produce yours. And you can as well make them as long as they can. Join the Guinness Book of Records. You can have one if you allow them to grow. Two, are we able to be in position to tell exactly how the process of transcription occurs? What does this other teacher do? How does he read out this decoded information? Tak, 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 tak. Letters are written in dots, but he has to make them appear as letters. Those who are doing ICT and IT, you understand what we mean when we say coding and decoding. The hackers. So, from that picture, we have three interesting words. One, the DNA. Two, RNA. Three, RNA polymerase. What does this suffix tell you? If this is DNA in purple, and then you have green as RNA, what's the difference? single or double strand? Why the opening? And why the closed ends? Is that where our coded information is read and then taken as a letter to be read and then, you know? Long time ago, they used to tell us that during colonial times, that a person in the village would be given a letter by the chief of the village to take to another office in the province. Master and all those. Now, so this person would take the letter stuck in between a paper, like this, and he goes carrying. So he takes it to the office, they read it and say, you know what, you're actually supposed to go to prison. So they read it for him and they check the instruction, this person carrying the letter is supposed to go to the prison, so you take yourself to prison because you can't read. Now, the tricky part here is, how is this information going to get out of the nucleus, go to the cytoplasm, and what shall it be containing that we should be able to see? That's only our focus today. We are not going to look at how proteins are made, but we are only going to look at how the information for the proteins is written and taken out. Simple. It ends where we have the information extracted. So, here we go. We need to remind ourselves of these three stages in terms of parts. One, the DNA, that is the gene. RNA as the message extracted from the nucleus by the process of transcription. So our focus, therefore, is here. So our next lesson is going to be here. So we, therefore, are moving from here, move, and continue only up to here. That's where we are. A copy of the gene used to encode a protein. The proteins we do have. And the diagram here helps us sum it up. Like we said, an overview. Here is your nucleus. This is your DNA double-stranded. Check what is happening and what pulls out and gets out of the nucleus through the nuclear pore. What happens in the cytoplasm is for another time. So, one, it's lengthy and it's variable. The average length in mammals is around these nucleotides. Oh, so much. Translating to about 730 amino acids in an average, check, average protein. So there could be some that are bigger than that. If you look at the molecule of hemoglobin, for instance, how many alpha proteins does it have in terms of amino acids in a chain? What about the beta ones? You looked at this in S4, so you still remember that. So only certain genes in a genome need to be expressed. Back to the previous lesson, gene expression, when our information is transformed into who we are. Then we're expressing what we have inside. Some people express their emotions and they express their anger by fighting. What about what we do have? One, that the way these genes are sent out as information and then we are able to see them as structures will depend on one. What is this cell doing? Is it specialized or it's not? Could it be depending on the environment? Which takes me to one interesting thing. Some people say, Africa, we are so unlucky, we never experience winter. You know, who would need it? Is there any good in it other than looking at it and say, I can also see snow. But what does the sun do to us? Is it the light rays from the sun that help us develop our pigment so well? Which takes us to one interesting component in our growth and development. Children, newly born, light skin, beautiful. Like, oh, I wish I was to remain with this skin color. Day 2, day 3, day 4. Kwitizina, munsurajezi. Bamuvanye mchumba, mjanye muri salo, then they start taking this kid outside of the house, in the compound. And then the skin tone starts to change. From lighter to chocolate, and then finally, midnight, black, African beauty, as you always talk about it, black skin. Remember one thing. That's a protein being made. Melanin, it's a pigment. But what accelerates its formation? Why is that when you bask in the sun for so much long, then your skin pigmentation kind of gets readjusted? And then you say, I don't look okay. Then you start going for mkorogo. That you want to get lighter. Remember what you're doing to your cells. These are chemicals. Can they be affecting your genes? Let gene expression be from within to outside of your body by itself. And also depending on the cell and the environment you're in. Not all the genes we do have are visible outside in terms of the structure they make. So we have inactive ones and those that are activated to be able to produce these proteins. So the process of transcription and translation, and then we have an expression, will be depending on what the cells do. We also have very many examples like sugar control. Why is it that some people are diabetic and others are not? Some have type 1 diabetes, which is inborn. We sometimes refer to it as hereditary because a child is born with it. And sometimes you find that some of those cases tend to run through families. And so we say, kind of hereditary. But essentially, we expect a normal human being to be able to produce insulin as a hormone to be able to control this sugar. So what does this mean? If I do not take so much sugar, would that mean that in my blood, the amount of insulin to control that sugar is always relatively low? After all, I don't have so much to break down or store and hide somewhere so I don't become diabetic. But the synthesis of these proteins, the expression of these genes to make proteins, will depend on the environment. Uriye umuceri n'ibishimbo n'imyumbati myinshi cyane. 1 hour after, your sugar goes high. And then minutes after, it goes down. Who is doing that? Is it the environment of the sugar, the environment around the cells, dictating some bit of signal? Hey, we are finished. Zana insulin vuba turashize, gabanya isukari. That when the sugar has gone down, then how much of that as insulin is going to be released after synthesis would also be readjusted. Which also takes us to tolerance, lactose tolerance. There are some people who are able to deal with milk, others say, I can't, you only give me needle, otherwise I would have constipation and stomach problems. Others say, I would rather take chivuguto, nacho chinwai, ariko amata y'inshushu, dekada, babari. Proteins depending on the environment, also expression depending on what we need. So, in one simple goal, what exactly is transcription and how does it connect to translation? Let's focus on the images here and this. Don't look at the words yet. Pictures are louder, remember that. So let's go together. That we do have a T, an A, and then C, C, G, A, C, A, G, T, onwards. And if I do have a T, check the colors. If you cannot read the letters, follow the colors and see. if we have any single color matching with another color of the same type? Are we talking about complementary base pairing? That the designer of the image says, you know what, if they can't see these letters, let me use the different colors. So I know that there is no purple going to meet another purple. What do you say? That there is no orange going to meet another orange. Let's check. If this is A, then this is A, this is T, and this is T. If we are to roll this back and fix it again, remember DNA replication we looked at the other time, that A with T, A with T, G with C, not G with G. Don't match the colors, match the letters. A for T and G for C. 2, that the opening, then we now have AU, GG, U, trace U, and see where it is. Now, a single strand carrying U instead of T, still in the nucleus of the cell. Once this has been extracted out, we refer to that as transcription. As simple as that. Extracting a copy of mRNA out of the DNA. But remember, this is a polymer as well. You remember that? A polynucleotide, RNA and DNA. Difference is double mass, twice the amount because of the 2 strands, and half the mass because this is a single strand. Now, the question is, once it is out, what name do we give it? Remember what we talked about before, the message. So it's a messenger for the DNA. So DNA itself does not leave the nucleus, but a copy of it in form of RNA will move out as messenger RNA. That the mRNA now travels from the nucleus to the cytoplasm. Check that very well. What it's going there to do, another time, but we can simply look at what is happening, that we now have our chains of beads of proteins of amino acids. And when you look at this is our region of the cytoplasm, and this is our region of the nucleus, meaning that we are only here and we are transcribing. Translation, which is here, making of the protein, the synthesis, the factory. So it simply means that this is the construction site, this is the map brought by the engineer. You'll never read it even if the house is your own. You'll have to wait for the engineer to say that, okay, in this section it was going to look like a brick and the building will look like this until he shows you a 3D, the expected building. You can never read his map. That's their specialty. A specialty for DNA is to transcribe and bring out our mRNA. Then what happens in the cytoplasm? Leave it for the ribosomes. All right. So when this actually happens, from the codes to airtime, mobile money, all those codes, they keep changing them. Have you ever tried to ask yourself why they keep changing those codes? And there are some people hacking them. They have to keep fine-tuning and adjusting according to the environment. As more people are born and people continue to get exposed to IT, they will hack until they go to automated machines and get the money out. And banks go losses. But moderately, we shouldn't be doing that. Just work and get paid for what you've done. But the question is our environment the same all the way through? Are we the same as we were born? And if not, why? We also need to ask ourselves, could these proteins being made be a kind of preparation mode to what is likely to happen, a long life? When do we need certain proteins and what are they for? Where are they coming from? Could it be that natural design has put in place all the information to cater for any changes that are yet to come, even in 100 years from now, amongst us as a species? Muscle contraction, cytoskeletons, blood clotting, membrane transport, all those are activities. But how do they happen? Where is the role connecting to structure that has been seen in a protein that was made through translation, having got the information from mRNA that was transcribed from DNA? Question. Why did our S1 teacher teach us that the nucleus dictates all cell activities? Why couldn't he tell us this well before 6 years ago? And here we are now. See, oh, so it is from transcription and then protein synthesis. Then I see red blood cells coming up, then they carry oxygen, and then I have respiration, then there are enzymes. Oh, so these are the characteristics of living things. Because the cell is a unit of life whose role is being dictated by the nucleus. There we go. So, to clearly go straight to something way back we looked at in DNA replication. Open the zip. Have two halves of the zip. But who is opening it? The section in the machine? What are we finally getting as parent strands? That in the unwinding, DNA is unwinding, but what enzyme do we have? And once this has happened, what exactly do we need? Think about it. That once the opening is over, the strands are separate. We need the energy. The bases that have been complementary have been separated. Once we've extracted the information, do we close the gap again? And once that is over, the question is, what exactly do we get? That from the DNA to the mRNA as transcription, what are the requirements? Is it the only mRNA we talk about? Which takes me to the very beginning when we were talking about nucleic acids. You remember? And we're talking about mRNA and then other types of RNA, which we said we are going to look at now. Here we go today. We have them now. That we do have one mRNA, which we have been focusing on, outcome, single strand, the sections you put in your hair. And here we go. But where does it come from? Could it be an extract from the DNA that's inserted in and then we extract it out and then it goes, and here we have a transcription? But do we need enzymes? Check that out. RNA polymerase. While we do have DNA polymerase in DNA replication, in transcription, we this time around need an enzyme responsible for catalyzing the process of formation of RNA. So, bottom line, the name of the structure made, and being a polymer, and ASE taking us to be looking at it as an enzyme responsible for that. So here it comes out, it's extracted, it's mRNA. But now the question is he the only one involved? Could we be having some others? Go to the construction site. Is it only the map for the building that is going to build itself? Do we have abakarani n'abaengineeri? Do we have trucks to bring sand? Do we have trucks to bring water? Are you going to put gravel and sand in a water tank that is bringing water to the construction site? Who is adding these bricks? So we do have 1, mRNA, as we said, 2, tRNA, transfer RNA. So we shall be comparing that to a truck that brings sand, gravel, stuff like that. Also, a small pickup to carry people, because where you put people, there is a seat belt. In a dumper, you don't have belts there. Ribosomal RNA, the primary component of ribosomes, that within the ribosome is another form of RNA. So the name is therefore extracted from the site in terms of location. Transfer, because it carries amino acids. mRNA, because it's the one with the message from where we have to read and make a protein. But like we said, we are only focusing on what? 2G. And there it is. The codes, the DNA, opening up, creating a section along which our RNA polymerase is reading off and adding the blue section. Now you need to check this, that for every strand we will ever meet that is for RNA, we shall always have U instead of me? No, U instead of T, as you can check. So it is the RNA transcript that is now finally extracted and expanded. Remember, it is a codon, sets of codons, so you have 3, 3, 3, 3, 3, 3, 3. So when we talk of anticodons, we are referring to other RNAs we shall look at. The bases here in the genetic code will be always referred to as those genes, and we do have the letter T always being replaced by U. Check where there is a T, there is a U down, and then extracted here. That if we had to look at this same line of arrangement, now extracted down, check it out. This would come back here when this is out. So we get the information and then zip again. Finally, would you do this for me? We talked about transcription, and there you go. See you another time as you wrap that piece